Hello students, welcome back to my channel Competitive Botany. In this video, I am going to give you a brief lecture on linkage. As you know that the genes are present in chromosomes. So chromosomes are the hereditary materials. They consist genes. So during the cell division, the two sister chromatids of a same chromosome, they reach the two different poles. So that's why the genes present in one chromatid, they reach the one pole of the cell, and the other chromatid, they reach the other pole. Okay, so as a consequence, genes fail to show independent segregation. That means they inherited together to a pole. So inheritance of genes together, or they retain their parental combination even in offspring, is known as linkage. So I will explain you easily, don't worry. So if you see where the genes are located, we all know that the genes are present in chromosome. Okay, the genes are present in the chromosome. On chromatid they present. We know that. So during the cell division, the chromosome, before the cell division, one chromatid will gives rise another chromatid with the help of replication. So during cell division we see in this situation. That means each chromosome containing two chromatids. These two are generally referred to as sister chromatids. They are referred to as sister chromatids. Okay, so during the cell division, these two sister chromatids, they are separated by a division of the centromere. Here the centromere is going to divide into two. Then it separates the two chromatids. Then one chromatid reach the one pole of the cell and the other chromatid reach the other pole. So then the two chromatids are separated. They reach the different poles. Okay, so whatever gene content present in the one chromatid will reach the one pole, and other chromatid will reach the other pole. That's it. So as a consequence, so these genes are, the gene content, whatever genes are present on single chromatid will reach the one pole without segregation. They tend to move exactly to the one pole. They do not tend to separate. So the whatever genes are present in the chromosome, whatever gene content present on the chromosome, they do not tend to separate during the gamete formation. Exactly, they went to one pole. Okay, so as it is. So this phenomenon is called as linkage. The genes present on single chromosome, they do not tend to separate during the gamete formation. They went to gametes as it is. Okay, so this phenomenon is called as linkage. That means inheritance of genes together, or they retain parental combination as it is in the offspring. That is called linkage. Okay. So let's see the another point. The tendency of two or more genes to stay together during inheritance is known as linkage. Two or more genes, they do not tend to separate. They went as it is to the another generation. So they tend to go as it is to the another generation. That tendency is called as linkage. Why? Because they are linked together. They do not tend to separate. So linked genes do not show independent as segregation. As a consequence, the dihybrid F2 ratio and the test grass ratios are significantly, we know that dihybrid phenotypic ratio is 9:3:3:1. Dihybrid test grass ratio is 1:1:1:1. We know that. But the parental character combinations appear much higher frequency than the recombinant phenotypes in the case of linkage. Okay, so these when the genes are under linkage, they do not tend to separate. We are not getting 9:3:3:1 ratio. Instead that we are getting more number of parental combinations and less number of recombinant combinations, as well as we are not getting 1:1:1 test grass ratio. Instead this we are getting their 7:1:1:7 ratio. So I will explain you later, don't worry. So the difference between linkage and independent assortment we can assume by two examples. We can know the two phenomena independently. Okay, let's see the examples. So let's see the first example. Genes located on different chromosomes assort independently, giving 1:1:1 ratio. For this I have drawn a classical example of dihybrid grass, that is round shape yellow colored seeds are crossed with wrinkled shape green colored seeds. When then we get F1 hybrid, that is having dominant characters. Why? Because recessive character will suppress in the F1 generation, only dominant character will appear in the F1 generation. So round yellow seeds were obtained in the F1 generation. So generally Mendel selfed them to get F2 progeny. So here we are going to know the dihybrid test grass ratio. For that we have to test F1 individual by crossing with a recessive parent. So let's see the F1 individual crossed with double recessive parent. This cross is called as dihybrid test grass. Okay, so according to Mendel, four phenotypes were obtained. They were round yellow, round green, wrinkled yellow. They were obtained equally. That means 1:1:1:1 ratio. This is dihybrid test grass ratio. According to Mendel, we get this ratio. Okay, so contrary to this, let's see the another example. So then linked genes do not assort independently. We already known that. Okay, but they tend to remain together. They do not tend to separate during the gamete formation. In that case, we are going to see the another case. Okay, so in this case, so the two genes were separated by a slash. Look at this. They were separated by a slash. RY genes. Why? Because I will tell you later. Okay, in detail. Okay, so let's see the cross. When the round yellow seeds were crossed with, that means the slash representing this is one chromosome, one chromatid or one chromosome, one homologous chromosome. This is the another homologous chromosome. This is one homologous chromosome. This is the another homologous chromosome. That's why we are representing a slash between them. Okay, so as usual we know that RY alleles will come into the gamete. Here also small r small y gamete will come. Okay, when they fuse together, we are getting, as you know that a dominant individual will be obtained in the F1. Okay, F1 hybrid is a dominant individual, that is round yellow. When we cross this to wrinkled green plant, then in the case of linkage, In the case of linkage, in the case of independent assortment, we should get 4 phenotypes, but here the phenomenon linkage is observed. That's why here we are going to get 1:1 ratio. That means this is the parental combination. Okay, this is the parental combination. Here also the parental combination. Only parents will come. We are not going to get any recombinants. Okay, we are not going to get any recombinants. This is called this is due to linkage. I will explain you in detail, don't worry. Okay, now we are going to see what is linkage. So the linkage was first proposed by the scientist T.H. Morgan. So before Morgan also, two scientists have independently proposed a phenomenon that is called linkage, but the credit goes to Morgan only. Okay, so let's see the Sutton hypothesis. Sutton, in the year 1903, he proposed each chromosome bears many genes, and all the genes situated in one chromosome, and they are inherited together in the offspring. This theory provides a physical basis for the heredity. So Sutton proposed that chromosomal theory of inheritance. That means the genes are present on chromosome, they inherit to the next generation of springs. So this theory is that is providing a physical basis for the hereditary nature. Okay, let's see the two important hypotheses. They are coupling and repulsion. Okay, so the phenomenon of linkage was first observed in sweet pea. So I already told you that before the Morgan, two scientists have proposed linkage phenomenon, but they named them as coupling and repulsion phenomenons. They didn't know the linkage phenomenon, but they proposed the same theories. Okay, they are Bateson and Punnett. They formulated two different hypotheses. They are nothing but coupling and repulsion. So to explain the linkage, okay, to explain the linkage only, let's see the first one, coupling phase. Bateson and Punnett crossed a homozygote sweet pea. Homozygous means homozygous. Whenever you saw homozygous, you should remember in your mind that is a pure plant. That is a pure plant. That means capital B, capital B, capital L, or small b, small b, small l, small l. Okay, when they crossed blue flowers, long pollen plant with red flowers, round pollen grain plant. So they got a dominant pair individual in the F1 offspring, that is capital B, capital L, small b, small l. Okay, so according to the law of independent assortment, F1 hybrid was expected to produce 4 types of gametes. We know that in equal number. So the blue colored flowers, long pollen plant crossed with red flowered plant, round pollen plant. Okay, so this is the dihybrid class. Why? Because we are considering two characters here. Okay, when they observed F1 individual, that is having blue flower, long pollen type. So F1 individual, we know that we get from F1 individual 4 gametes will come to form F2 progeny. So when F1 individual is backcrossed with a double recessive parent, that means red and round pollen parent. So here also we are going to get 4 different types of phenotypes. They are blue long, blue round, red long, red round. Okay, so but here the situation is different. So instead we are getting 1:1:1:1 ratio. Here we got 7:1:1:7 ratio. So have a look. Why we are getting more number of parental combinations? Have a look. This is the parent. Okay, isn't it? This is the F1 parent. This is also double recessive parent. Okay, so parental combinations we get more number, and recombinants, these are the recombinants, we got less number. Why this is occur? Why? Because I will explain you 1 minute. So have a look. This is the first parent, that is blue long parent, and this is the second parent, that is uh, blue red round. This is blue long red round. Okay, have a look. Why we keep a slash in between capital B, capital L, small b, small l? Why? Because the two genes, the two different alleles for dominant character, they were present in the one chromosome. The two small character, two recessive characters, that means represented by small letters, they were present in the different chromosome. These two are the homologous chromosomes. We know that they were in pairs in us. Okay, so one we get from the father, one we get from the mother, we know that. Okay, so here the situation is this. That's why we kept a slash in between them. That means these were present in one chromosome, these two were present in the other chromosome. This is called as coupling phenomenon. Coupling means, so these two are coupled to each other. Okay, they were present in the same chromosome. As we know that the genes present in the single chromosome, they do not tend to separate during the formation of gametes. That's why they went as well as same to the next gamete. Okay, they also, this chromosome also went to the gamete as it is. That's why we get more number of parental combinations. Here also the same. So the gametes with BL, they are more in number. They are formed more in number. That's why here we got a more number of parental combinations. So what are the you have to know what are the recombinants. How we will get recombinants from these? So if the crossing over occurs in between these two genes, we get recombinants. How we get recombinants? Let's see. So capital B, small l. So that is the recombinant chromosome. I will draw. Capital B and small l is the recombinant. Another one is small b, capital L. This is also recombinant. Okay, by a crossing over is another phenomenon. I will explain you in the another class. Okay, so these are the recombinants. So we are getting these recombinants but in less number. Okay, we got more number of parental combinations. That's why we got blue long 7 and red round 7 recombinants in only 1:1 ratio. So that's why the ratio is instead 1:1:1:1 ratio, we get 7:1:1:7 ratio. Okay, this is the coupling. Now we are going to see the another situation, that is a second situation, that is repulsion phase. This is also proposed by Bateson and Punnett. They were taken two plants such as the different plants different from above cross a coupling phase. Okay, look at the genotype. Okay, so capital B, small l, capital B, small l, small b, capital L, small b, capital L. That means the phenotypes are different. That means one dominant gene, one recessive gene is present on the same chromosome. Should remember. So first one is blue round, second one is red long. Blue flowers, round pollen plant is crossed with red flowers, long pollen plant. So then as usual we know that the dominant character will come in the F1 hybrid. We get blue long. blue flowers, long pollen. So blue flowers, long pollen will gives rise 4 types of gametes, and it is going to back crossed with the recessive parent, that is BL and BL. So from this we get only one gamete, that is small b small l. Okay, here the results are same. So in coupling phase and repulsion phase we are getting the same results, but the case is different, you should remember. So blue long, blue round, red long, red round. Here also we got 7:1:1:7, but the situation is different, should remember. So the parents are different. We should there we got in the coupling phase we got more number of parental combinations, but here we are not getting more number of parental combinations. Actually these two are the parents here, these are the recombinants here, but here also we are getting recombinants in more number, but the parents in less number. Why? Because so these are the parents taken in the cross. Okay, so same chromosome consisting one dominant, one recessive, one dominant, one recessive. So this kind of arrangement is called as a repulsion arrangement. That means when the one dominant, one recessive gene present on the single chromosome, they tend to separate. Okay, that means they are showing repulsion. That means they wanted to separate themselves during while going to the gametes. That's why here we got a more number of recombinant. What is the recombinant here? What is the recombinant here? Okay, so the recombinant here is, so when the crossing over occurs in between these two chromatids, then BL is here. The BL is the recombinant. Small b small l is the recombinant. Okay, so that's why we got a more number of individuals, more number of recombinants here. Okay, so this is the case different from the above case. In the coupling phase, we got more number of parental combinations, but in the repulsion phase, we got more number of recombinants. Okay, so these two are the recombinants here. Okay, these two are the parental combinations here. Should remember. Okay.